So I just gave you a small introduction of the process of transcription and translation. And now I'm going to talk about how scientists crack the genetic code and why the genetic code is redundant but not ambiguous. Around 40 or 50 years, we didn't know what were the rules that allow the assembling of amino acids into proteins that are encoded into DNA. We knew that there were 20 amino acids, but there were only 4 nucleotides in DNA. So we didn't know how many bases correspond to an amino acid. Now we know that there are 3 nucleotides per amino acid. And in class, maybe, and also here, you will learn how to use this table in case that you don't know. I'm sure that many of you know. So I told you 3 nucleotides will correspond to an amino acid. So in this table, it will be 1, here 2, and let's suppose that this will be, if you have the nucleotide G, C, and G, you will have this amino acid that is alanine. If you have, for example, A, and A, you will get lysine. Now we will talk about the relationship between translation and codons. And to start talking about that, we have to start talking about the template strand. If you remember, the DNA has 2 strands, and to copy the DNA or for replication or for the synthesis of RNA, you have to open the strands, and one of those 2 strands will be the template strand. So you will determine the ordering of the sequence of nucleotides. So based on the template strand, you will say this will be an A, C, C, and they will go first. The second thing that I want to tell you is about this structure that is very important, codon. And if you see, the codon is formed of messenger RNA and is 3 nucleotides. So it's a triplet. And here you can see the first nucleotide, the second nucleotide, the first codon, the second codon here, third codon, and this is the fourth codon. And the last thing that I want to tell you is that based on these codons, you know which amino acids will be located first or last. Yeah, so the position of the amino acids depend on the order of these codons. So let's recap all the things that we have talked. I told you that during transcription, one of the 2 DNA strands, they call the template strands, provide a template for ordering the sequence of nucleotides into the RNA transcript. So I told you that, for example, this DNA strand will tell you that first will go an A, then it will go a C, then the third one will be another C. The second thing that I told you is that during translation, you have messenger RNA-based triplets, these ones, that are called codons, and that are read from a 5' to a 3' direction. So here, 5' to a 3' direction. And finally, that each codon specifies the amino acid to be placed at a corresponding position along the polypeptide. So that this one is first and this one is last, depending on the codon that you have here. So as I just said, if you have A, G, T in your template strands, you will form a codon of that is will be U, C, A, and then an amino acid that in this case will be serine. And if you see, the flow of information from genes to proteins is based on a triplet code. So all the diversity from bacteria to different vertebrates going through insects, All of them use the same thing. All of them use these triplet codes, and this is the smallest unit of information. So although we are so different, all use the same mechanism, that is this triplet of bases. So let's recap all the things that I just said to you. So I told you that the flow of information goes from gene to protein and is based on a triplet code. And that is a series of non-overlapping 3 nucleotide words. This is the first thing that I want you to remember, and that these triplets are the smallest unit of uniform length that code for an amino acid. And I show you this example that produce serine going from template strain to codon and then into an amino acid sequence. Now we're going to talk about DNA complementarity, and we already talked about this, so we know that A will always pair with T, and I use my keyboard as a mnemonic to remember that A and T go together. Also G and C go together, and I remember the symbol of Geico. You can use this mnemonic, or you can use another mnemonic, or not use mnemonics. The important is that you remember them. And for example, you can answer here based on that. What is the DNA complementary strand? So for example, here it will be T, because we know that A and T go together. Here we know that T and A also go together, so this will be an A, and you can continue doing the rest. Okay, now that you did some of these ones, now I show you the answer. This will be C, T, and I'm sure that you are able to do this. Maybe there will be a few examples in class and or some of them in the test. In the same way, to go from DNA to RNA, you know that there is a small change in. The T is replaced by a U, and again, I use mnemonics, so the Geico symbol still work. However, I use now aura as a way to remember that A and U always are go together in the RNA. So now let's try to go from a DNA template strand to an RNA template strand. And you can use again these mnemonics, or you can use something else, but I want that you stop the video and try to answer these questions. Okay, so now you know that A and U go together. So you show here, A and U go together. Now the T, now we don't have T, but the T was replaced by the U, so this will be an A. And G is going to still be the same. So G and C go together. And you can continue like this in all of the examples. In the middle of the 1960s, they were able to crack the code, and they were able to see that there were 64 triplets, and those 64 triplets, they were divided into 2 groups. 61 code for amino acids, and 3 code for stop codons. And what is a stop codon? Stop codon is a codon that say to the protein, to the process of translation, stop. This is the end of this protein. But if you see, this is very interesting because instead of having one, they have the same, they have 3 codons to send the same signal. And if you think about the 61, as we said before, there were only 20 amino acids. So you have extra copies here for both. So they found that the code has 2 properties. That the code was redundant, and I will talk in a second about that. However, the code was never ambiguous. So yes, for redundant extra copies, but you always are going to get the same amino acids, and I will talk about that in the next slide with an example. they also found that the reading frame is very important. So for example, if a mutation appears that removes this nucleotide, now you're going to have a completely different codon. So now this will be 1, 2, 3. So this will be a different codon. This will be a different codon. This will be a different codon. And this will cause that all the amino acids were different. So the reading frame is very important. So now let's try to go and recap all the things that we said. So I told you that we were going to talk about these structures that are the codons, how they crack this code. And I told you that they around the 1960s, in the mid of the 1960s, they found that there were 64 codons. However, from these 64, only 3 triplets encode for a stop codon that stop the translation, and 61 codes for amino acids. So as a result of that, they found that the genetic code was redundant. However, is never ambiguous. So no codon is specified for more than one amino acid. And again, just to finish, I told you that codons must be read in the correct reading frame. And I give you an example that if you remove one nucleotide, you will remove the reading frame of the codon, so you will get completely different proteins. Now I want to explain in more detail what is the difference between redundant and not ambiguous. So here is an example in which you can see that the genetic code is redundant because you have 2 codons that encode the same amino acids, and here you have another example. And if you see here, you have even 4 coding the same amino acids. However, you will never see a codon that code for more than one amino acid. So it's not ambiguous, but it's redundant. And here I also wanted to show you, here you can find the 3 stop codons. Now I want to explain in more detail what is changing the reading frame. And to do that, I have 3 frames. In the first frame are comparing all this RNA sequence. Now in the second frame, you change the reading frame and you remove the U. And as you can see, you have completely different amino acids. Yeah, all of them are different. And if you remove 2, so instead of the, now you have the U and you don't have the C, you again are going to have a completely different sequence of amino acids. And this is very important, the methionine, here it is, because this is the start codon. So all the proteins are going to start with this start codon, and they're going to finish with this stop codon. So this, the third frame, is actually what is more realistic, what you will find in nature. You always are going to start with the methionine codon, and then you will finish with one of the 3 stop codons.